Hi there, this is FemmeSciComm's 11th vlog, and to segue from last week's post, today's post is on PCOS. PCOS can be defined as hyperandrogenism and or chronic anovulation, which may lead to infertility. Cysts are present inside of the ovary, and coinciding metabolic issues can occur, such as insulin resistance and obesity. Sadly, the cause or causes of PCOS haven't been identified or described yet. Today's paper looks at possible factors in PCOS pathogenesis. So this paper was recently published in Molecular Medicine Reports, Bioinformatics Identification of microRNAs involved in polycystic ovary syndrome based on microarray data. The first author is Yan Hu, and the senior author is Shuqing Wu, both from the Second Hospital of Shanxi Medical University. This study tries to answer which microRNAs are involved in PCOS. First, what are microRNAs? MicroRNAs are small, non-coding RNAs of 20 to 22 nucleotides in length that are capable of regulating protein expression and other cellular processes. Because not a lot is known about PCOS pathogenesis, this research group conducts this exploratory study by casting a wide net to determine if any microRNAs are dysregulated in women with PCOS. And further down the line, narrow down any biological processes that are targeted by these microRNAs. So onto their data. When looking at the characteristics of the women in this data set, the researchers found that women with PCOS have a statistically significantly higher BMI compared to women without PCOS. In addition, the levels of their hormones LH and FSH are also different compared to women without PCOS. LH is higher and FSH is lower. This has already been noted by medical doctors, but what causes this hormone level change hasn't been fully described yet. 7 microRNAs are differentially expressed in granulosa cells of women with PCOS. These 7 microRNAs are MIR3188, MIR4433 3P, MIR3135B, MIR1587, MIR1225 5P, MIR4417, and MIR4749 5P. Via bioinformatic analysis, the researchers identified that these 7 microRNAs target 935 genes. These 935 genes have functions such as homophilic cell adhesion via plasma membrane adhesion, cellular response to nitrogen, myeloid cell development, and regulation of cell projection organization. Again, using bioinformatic analysis using the FunRich software, the researchers identified that these microRNAs can regulate the following transcription factors: transcription factor SP1, myogenic factor 5, homeobox protein NKX 2.1, nuclear factor 1 C-type, homeobox protein NOBOX, early growth response protein 1, RAS responsive element binding protein 1, visual system homeobox 2, T-cell acute lymphocytic leukemia protein 1, and central areolar choroidal dystrophy. The ones highlighted here are statistically significant. The research group validated their findings via RT-qPCR. They identified 2 out of the 7 microRNAs are differentially expressed using their cohort of 73 patients. These microRNAs are MIR3188 and MIR3135B. MIR3188 has been implicated in promoting tumor cell proliferation and migration. MIR3135B may aid in oocyte maturation in the ovary. So what does this all mean? This is a preliminary study that identifies potential biological processes that are dysregulated in the ovaries, specifically the granulosa cells of women with PCOS. There are several theories regarding the cause or causes of PCOS, and this study adds to the growing scientific literature that faulty gene regulation may be to blame. MicroRNAs are a form of gene regulation and may potentially be a target for PCOS therapies. The main treatment option for women with PCOS is birth control. But for women with PCOS that want to have children soon, this isn't really a viable option. Focusing on other aspects of the PCOS disorder may be a better therapeutic option for these women. there are caveats and factors to keep in mind. The processes and transcription factors the authors think these microRNAs regulate were not validated in this study. Functional studies are needed to determine if they truly play a role in PCOS. Also, this study uses granulosa cells, but there are several types of cells present in the ovary: theca cells, germ cells, oocytes. Other research groups believe that the theca cells are the original cause of PCOS, but it's still uncertain which cell population is more important. Overall, this preliminary study pinpoints 2 to 7 microRNAs that are differentially expressed in the granulosa cells of women with PCOS. The expression of MIR3188 and MIR3135B needs to be validated and their functions detailed in order to identify them as important factors in PCOS pathogenesis. The identification of these microRNAs and the genes that they regulate may provide a better understanding about PCOS and may aid in the development of additional therapeutic options for women with PCOS. Thank you for reading, listening, and watching my assessment on this paper on PCOS and microRNAs. Keep informed, stay interested, and keep doing great women's health research. Thank you for watching. If you're interested in this content, please make sure to subscribe to my channel. Check out my website, FemmeSciComm.com. I post every Thursday on new research papers pertaining to women's health. You can also follow me on Facebook, Instagram, Twitter, and Pinterest at FemmeSciComm. Bye!